Hello everyone, and welcome back to our training series on protein model quality. Last time, we explored the fascinating world of predicted protein structures, specifically the application of AlphaFold's pLDDT score to alignments and 3D structures alike in Jalview. In this part of the series today, we will consider the predicted aligned error, another measure of model uncertainty provided by the database, and some very basic ways in which we can explore this further. Let's start with an example that is native to all of us. Calmodulin, a protein that controls the transport of calcium ions during synaptic transmission in the brain, a process we're all relying on as we work through this video. Here, I've built an alignment of calmodulin in Jalview using the GL-props alignment algorithm for sequences expressed in the yeast, social amoeba, roundworm, cow, and human species. Next, I'm going to search for predicted models using the 3D Beacon search facility we have in Jalview. And we've got experimental and predicted models from all the providers listed together. So I'm going to refine this list so we have models by AlphaFold only featuring at the top. And now, I'm going to load these into the configured 3D viewer, which is Chimera X. Just to bear in mind, we also have PyMol, UCSF Chimera, and JMol available in Jalview for the purposes of 3D visualization. Great. First, I'm going to hide all default alignment-related annotation we have featuring up here, so we have more clarity. Now, the predicted aligned error can be extracted and loaded as a reference track, along with the secondary structure and the pLDDT score, also extracted from the AlphaFold database. This is a simultaneous process that happens for each sequence in the alignment where data is available. You'll notice that these are quite compact at the moment, but they can all be enlarged using the same mechanism, which is holding the Shift key whilst click-dragging one of the tracks. And this replicates the effect for all the other tracks of a similar nature. So, what is the predicted aligned error? Essentially, it is a measurement between all pairs of amino acid residues constituting a protein. And this is what makes it different from the pLDDT, which is computed for each residue separately. Dark green in this plot represents smaller error, and a greater probability that the two residues for which it was computed have relative positions that are well predicted. Now, the PAE plot for calmodulin in yeast here shows two rectangular regions. This suggests that there might be a domain at either end of this model's central helix, but it doesn't suggest whether this helix is rigid or inflexible. These putative domains can also be observed in the AlphaFold structure, loaded here in Chimera X. Now, Jalview provides a tooltip on the PAE matrix with the row and column numbers of the residues as well as the mean PAE measurement specified. Clicking on a point on the matrix highlights the residues that the pairwise measurement was computed for, up here in the alignment window. And if you want to make a rectangular selection, simply press down on your mouse's scroll wheel, and this will again highlight all the corresponding residues in red. To do this using just your keyboard, simply hold down the Alt or Command keys and click-drag to select the desired region. And that covers the basic functions of extracting, visualizing, and interpreting the predicted aligned error in Jalview. In the next part of this series, we will be looking deeper into the interactions between protein domains by clustering the predicted aligned error scores in Jalview using another feature. Till then, if you want to learn more about the 3D Beacons facility featured earlier in this video, Don't forget to check out our training video on the topic. Thank you for your time, and I'll see you again very soon.